Hey guys, quick biochemistry basics here. Let's talk about Sanger sequencing method. Now, before moving further, this video is divided into three parts. The first part explains the principle of the method. The second part is about classical Sanger sequencing that was used widely in 1980s. And the third part is about modern Sanger sequencing that uses fluorescence dyes. Sanger sequencing is based on dideoxynucleotides in DNA polymerizing reaction. Let's understand what are dideoxynucleotides. The nucleotide has three components: the nitrogenous base, which can either be A, T, G, or C, the sugar residue, and the phosphate attached with the sugar residue. When all three components are present, it's called nucleotide. If the phosphate is absent, then it's called nucleoside. Now watch carefully. If the hydroxyl group at the 2' end of the sugar is removed, then it's called deoxynucleotide. The deoxynucleotides are present in the DNA and they are used as a substrate by the enzyme DNA polymerase. All the DNA polymerase, whether it's DNA polymerase 1, 2, 3, Taq polymerase, etc., uses DNTPs as their substrate. The hydroxyl group attached at the 3' end plays a very important role in DNA polymerizing reaction. During DNA polymerization, there is a reaction between 3' group and the phosphate of the new DNTP. In 1977, Frederick Sanger had a brilliant idea. If the 3' group of the nucleotide is absent, then there won't be addition of new nucleotides and the reaction would stop. The nucleotide having 3' hydroxyl group absent is called dideoxynucleotide. Di means two and deoxy means absence of the hydroxyl group. Let's see classical Sanger sequencing method. The template DNA to be sequenced is divided into 4 tubes. Each tube has a primer that will bind to the template DNA. All the 4 DNTPs, that is the ATP, the TTP, the GTP, and the CTP are present. Along with these, DNA polymerase enzyme is present that will add DNTPs on the template DNA. Now here comes a very interesting part. This reaction mixture also has one radiolabeled dideoxynucleotide at a very low concentration. What I mean by this is that tube 1 will have dideoxy ATP, tube 2 will have dideoxy TTP, tube 3 will have dideoxy GTP, and tube 4 will have dideoxy CTP at a very low concentration, and all of them will be radiolabeled. Now let's see what happens in tube 1. The DNA polymerase would add DNTPs on the template DNA and carry out normal polymerization reaction. If somehow the dideoxynucleotide is incorporated, then the reaction suddenly stops. The reaction terminates as there is no 3' hydroxyl group for the addition of new DNTPs. The incorporation of dideoxynucleotide and termination of polymerizing reaction is a random process. Hence, the overall mixture will have several fragments that has experienced chain termination. Now, if the length of the fragment is known, then the location of adenine in the DNA can be estimated. The length of the fragments can be obtained by performing polyacrylamide gel electrophoresis and autoradiography. Once the autoradiogram is obtained, we can now have the sequence information of the desired DNA fragment. Okay, the first nucleotide is gonna be A. The second nucleotide is A, third is G, fourth is C, fifth is T, sixth is T, seventh is A, eighth is G, ninth is C, and 10th is again C. Now let's see modern Sanger sequencing method which is used even today. Now in this method, each of the dideoxynucleotide is attached with a fluorescence dye. The fluorescence dye can either be yellow, green, blue, or red. Instead of having an X-ray film placed on a gel, we'll now have a gel that would give fluorescence when exposed to UV light. As each of the dideoxynucleotide have their own fluorescence, it is now possible to carry out the reaction in one single tube rather than 4 different tubes. The chain termination reaction would result if dideoxynucleotide is incorporated in the DNA. The chain-terminated fluorescence fragments can be separated by polyacrylamide gel electrophoresis in a single well. The more advanced system uses capillary electrophoresis for the separation of fluorescence DNA fragments. These capillaries are further equipped with CCD or charge coupled device for the detection of fluorescence signals.